So, Helmut Hillebrand here, University of Oldenburg, together with Miriam Gerhardt and Marien Striebel, who are also here. We are happy to welcome you all to this presentation by our dear guest, Mary O'Connor, whom I will introduce in a short while. Before I do so, some of you will already have heard this. The meeting is recorded. After the talk, we have the chance to ask questions, but only via the chat function, which is in the bottom panel of the Zoom meeting. There's this chat symbol. There you can post questions. We will read these questions, and Mary will have the chance to answer them. With that, I would like to introduce Mary O'Connor, professor at the University of British Columbia in Canada. Mary and I share a number of research interests, and we have sat together in real rooms before. So this is a new version of interacting also on this plane. Mary got her bachelor degree from Brown University and did her PhD at the University of North Carolina, Chapel Hill, after which she did a two-year postdoc at NCEAS, the National Center for Ecological Analysis and Synthesis, also one of my favorite places on scientific places on this planet. And after that, she became professor at the University of British Columbia, where she still is. A few years ago, Mary gave a presentation in Oldenburg as the Edward O. Wilson Lecturer, which is a once-a-year lecture, and it was a lecture on the different aspects of information in ecology, sensing information, and how information might play a role in structuring ecological community. Today, Mary will speak about considering variability as a biologically important aspect of global change. And I'm very much looking forward to your presentation. Mary, the floor is yours. And I made a mistake. The question should be asked via the question and answer button at the bottom row of the Zoom meeting. Sorry for that. So Mary, please.
Okay, thank you, Helmut, for the introduction, and Helmut, Marien, and Miriam for the invitation. I think this is a really interesting idea, and as I said before, a great way to come together at this time when we can't really come together. So, and it's just always a pleasure to reconnect with you and your group. So yeah, today I'm going to talk about variability, and I was excited by this invitation, perhaps against my own best interests, because I've never actually given a talk on variability before. But the reason I was interested to think about this is that, as with probably any ecologist, any experiment I've done has had a lot of variability in it, and in particular, variability in the factors that I'm most interested in, like temperature or biodiversity. So I'm excited to be doing this, and I understood the invitation and the purpose of this webinar to be an incentive talk, which is to say I was asked to offer some ideas on variability to stimulate thinking and discussion, in particular about mesocosm work and experiments. So these are just a few things from the notes. And I mentioned this at the outset just because what I decided to do was to do exactly this, some ideas and some things that maybe I won't fully resolve in the talk, but I hope that they get us talking and thinking afterwards. So it's a little bit of an unusual seminar, but I really like it. Okay, so these are the topics. So like we all know, regular variation or variability is a defining feature of most aquatic ecosystems. These are the three that I work in and the ones that I think about when I think about ecology: lakes, seagrass systems, and rocky intertidal systems. And of course, rocky shores experience famous and notorious variability on a daily basis with the tides, as well as seasonality. Seagrasses are highly seasonal systems, even though we're coming to understand that a bit more only recently. And lakes are famously structured by seasonality in abiotic and biotic processes. And we know a lot about these three systems because our group, but also many groups, use mesocosms to study them. So these are just some pictures of our usually pretty low-tech mesocosm systems. And I'll get into later some of the advantages and opportunities that mesocosms can provide for understanding important ecological processes, for understanding global change, and in particular for thinking about variability. So in our research, we are almost always manipulating temperature as the independent variable, and we're almost always measuring community and ecosystem processes on the y-axis. So I just mentioned that so that you know where I'm coming from when I'm giving examples and thinking about things. As we all know, temperature is not the only factor that can vary a lot in nature and that can structure ecological patterns and processes. And it could be that the variability in other factors like pH, light, nutrients, and precipitation have perhaps different effects on ecosystems than temperature. So the outline for the talk is I just wanted to briefly touch on this idea that variability is in fact foundational to diversity, organization, and complexity. It's not only something we think about with global change. So just after a few words about that, then I'll spend most of the talk giving some examples from our work about how variability, and specifically considering variability, changes our predictions about change in ecological systems. I have a couple of examples really that hone in on the variability in nonlinear processes through both ecological and mathematical mechanisms. And then a totally different perspective on variability, offering a framework that we've recently come up with to help think about this. And then I'll end with a few words specifically on mesocosms and their opportunities and challenges for this problem. Okay, so life exists in a variable world. Just a couple of examples, seasonality in aquatic, coastal, and freshwater systems famously influences the primary production, the timing of it, the peak of it, as well as a secondary production through spring blooms, fall blooms, and transitions in grazing pressure. And then on land, so this is primarily driven by abiotic variation associated with seasonality and latitude. We also know that variability can come from biological processes. So population dynamics internal to one species or the connection between the two, as is famous in the lynx hare example at high latitudes, where oscillations in populations create variability in the environment that other species and other parts of the ecosystem need to respond to. So we can have biotic and abiotic drivers of variability. Now, actually, variability is so fundamental that some people would argue that life exists because of variability. Specifically, energy gradients represent changes from one place to another or one time to another in energy content. And one definition of life is that it's the harvesting of energy along gradients to catalyze metabolic reactions. So we could say that life exists because of variability in the environment. In a thermodynamic zero world, there would be no little variability, no structure to it, and maybe there would be no life. Furthermore, we know very well that diversity exists and persists because of variability. If the world were abiotically homogeneous, some of our theories suggest that we would not have diversity. We might have a superspecies, or at least dominance by several phenotypes, harvesting resources. And there's more recent theories suggesting that, well, maybe if there was no abiotic variability, we would have a few different types of life. But actually we would have a lot more because of the biotic interactions between different life forms would create additional variability in the environment. So there's hypotheses from biotic and abiotic corners that explain diversity in very simple terms, and its persistence. Furthermore, we don't get competitive dominance when the environment is variable. There we go. So variability, we can kind of sketch out a conceptual cartoon of this by suggesting that variability in the abiotic environment affects diversity. We know it leads to diversity. We also know that variability in the abiotic environment, let's say fluctuating temperature or changes in resource, can lead from a bottom-up perspective to changes in productivity, changes in biomass. But diversity actually can minimize those fluctuations, the biotic responses to abiotic variability. So we can get a feedback here where variability leads us to diversity. It allows the persistence of diversity, which then in turn actually stabilizes other responses to variability. So that gives us a diversity productivity relationship that's positive. So community and population ecology are really full of and based on feedbacks between variability and ecological structure. And I think as we move forward to think about global change and changes in variability, I actually think we still have a lot to understand. Even though our foundations are on variability, I think we can do more to be thinking about the role variability plays in the ecological systems we know before we even think about global change. There's a lot more to do there. And this is where a lot of our mesocosm work is now, actually, as we're starting to introduce variability into these systems and into our questions, we realize we're kind of starting at the beginning here. And that's okay, that's exciting. And I think it will actually strengthen our global change science to ground ourselves in this sort of thinking. But as we know, nearly all of us are actually engaged at some level in making predictions about the future. Any experiment that I'm part of or that I'm aware of is done in the context of global change and a changing world. So it is important to be thinking both about fundamentals and about how they lead us to predictions. So I'll give you a couple of examples from our work here, one on diversity and one on temperature. And the first one is on variability in nonlinear processes, part one, specifically Jensen's inequality and diversity in seagrass communities. And I wanted to just say here, I was thinking, unsure of exactly who you would be in the audience. And I know that for people who have started to work on variability as a major focus, Jensen's inequality is definitely probably front of mind. It's something that a lot of people are thinking about recently. But if you are not yet working on variability or thinking about it, Jensen's inequality is perhaps one of the most important concepts to really understand early on. So I thought it was worth including here, just to make sure we're all starting with that kind of explanation in mind. So this first example comes from seagrass communities. Seagrasses are found throughout, this specific species of seagrass, Zostra marina, is found throughout the Northern Hemisphere, and seagrasses are found throughout the world's coastal oceans. And the interesting thing to me, and for the focus of our work, is actually the animals and the high levels of biodiversity from invertebrates to vertebrates that live in eelgrass meadows. So we'll be talking about biodiversity, particularly of this guild of the small invertebrates. So seagrasses form patchy environments. So this picture is just showing you some islands near Vancouver, and the green blotches are the seagrass meadows mapped onto these islands. So you can see they form patchy environments. We know that the animal communities, or we believe the animal communities, are highly connected by dispersal, so movement among these patches. And we also know that species interactions matter on a local scale as well as abiotic processes. So we are really starting to think of seagrass communities not at the patch level, but at the meta community level. And this means, of course, that local conditions for biodiversity have regional consequences, and regional conditions have local consequences. So when we think about dispersal in particular, and we're getting into our first nonlinear process here, is the relationship between dispersal and diversity. So we know that as dispersal rates increase, as you go from no dispersal to very high dispersal, you get a hump-shaped or a unimodal pattern where local diversity in a patch is going to increase as dispersal rates increase because more species can get there, and they can be, the diversity would be low here because of either competitive exclusion or just lack of colonization. And at high levels of dispersal, again, you can get competitive exclusion by a dominant species that gets everywhere. So in the middle, you have a sweet spot of high local diversity. Local meaning alpha diversity. We can also think about beta diversity, and this will be the focus of the next few slides. So when you have no dispersal, we expect beta diversity or species turnover to be the highest in this region because each patch is left to its own dynamics, and we're likely to see different species dominating in different patches, either through deterministic or stochastic reasons. And as dispersal gets higher and higher, again, you get the dominant species able to get everywhere and dominate, so beta diversity becomes very low, less species turnover. So we're going to focus on this nonlinear relationship first. So Jensen's inequality is the term that simply describes the relationship between the mean of a function and the function of the mean for nonlinear functions. So this is a figure, the example I'm giving comes from a recent paper led by Adrian Steyer, and it was out in Marine Ecology Progress Series. This is a figure from that paper to illustrate Jensen's inequality for dispersal. So on the x-axis, just as in our cartoon, we have low dispersal rates going to high dispersal rates. And beta diversity, in the blue here, we would see beta diversity of high dispersal would be very low. Beta diversity of low dispersal would be very high. If we were to consider an average dispersal rate, we would get a beta diversity here sort of in the middle. But if we consider that dispersal rate varies, as I'm sure it does in something like a seagrass system where you have weather events, you have oceanographic events, or population dynamic events that would lead to sometimes having high dispersal and sometimes having low dispersal, the average dispersal rate, even if with the same average dispersal rate, if we allow variance in that, our estimate of beta diversity is actually going to go way up. And this is because of Jensen's inequality, the fact that taking the average dispersal rate with no variance gives us a very different answer than the average with variance. Okay. So when we consider variability in dispersal rates, we predict higher beta diversity than we would if we just consider a single constant dispersal rate. And right now, in most of the simple sort of applied models that we make, or conceptual models, and the experiments we do to test them on dispersal, we use constant dispersal rates. We use low, medium, or high, but we don't allow them to vary because we're trying to clean up sources of variation to test our hypotheses. But in this study, we explicitly looked at variability. So Jensen's inequality provides one hypothesis for what we should see if we have variable dispersal rates in seagrass. We should see high beta diversity, and specifically higher than we would expect. There's an alternative hypothesis, which is an ecological hypothesis. And this came from a nice modeling study by Matthias et al. In 2013 in Oikos. And here we see the same types of graphs I've shown you already with local diversity and beta diversity as functions of dispersal. And for beta diversity here, because there are different metrics, I'm just going to remind you that on this graph, more different communities are towards the top. So communities are very different with low dispersal, they get more similar. So the important thing here is they did some simulations where they looked at increasing dispersal stochasticity and then remodeled these relationships. And so as stochasticity, so in this case, random variation in dispersal increased, the effect of dispersal on local diversity got weaker and overall diversity declined. So we see a pretty strong effect of variability in dispersal on the way we understand dispersal and diversity to be related. And I'll just mention again, this hump shape is really central to a lot of our meta-community and meta-population models. So we're showing that variability actually can really weaken that relationship. So increased stochasticity and variability in dispersal, reduce diversity and reduce the effects of dispersal and diversity. Those were their findings. And their explanation in terms of their model is that shifts in dispersal affect the importance of competition. So remember, this hump shape comes from the balance between competitive exclusion and dispersal limitation. And they were saying as we shift, as the variability in dispersal carries out and the effects happen, we're seeing a change in the effect of competition. So it's an alternate hypothesis, an ecological hypothesis, in contrast to Jensen's inequality. So we tested this experiment with we tested these hypotheses with data from an experiment that had been done and published in 2009 as a part of Sarah Lee's PhD thesis. And this was looking at saturation and dispersal variability in communities. This is an excellent paper that I encourage you to check out. And then we reused the data because Adrian realized they would be suitable for this question about variability. So here's what we found when we reanalyzed those data. So we found for beta diversity first, this was the hypothesis, as supply rate here means dispersal from no dispersal to high dispersal. In the low variability treatments, there was really no effect of dispersal on beta diversity. So that's counter to this hypothesis. Maybe we're already out in this part of the curve. And for high variability dispersal rates, we saw generally greater beta diversity and maybe more of a suggestion, though not significant, of these of that relationship. So here we reject Jensen's inequality as the main driver in the patterns that we see, and the reason is first off, we just don't see clear evidence of this hypothesized nonlinear relationship, and Jensen's inequality relies on this nonlinear relationship. So our interpretation here is that we suggest that there is support for the shifting strength of competitive exclusion to explain how diversity and variability in diversity can affect beta diversity. And just in case you're interested, here are the results for local diversity. They are, again, perhaps suggesting we're on just part of the curve. We don't see the unimodal relationship with the dispersal intensity increases, but we do see an effect of variability as we would expect, consistent with the Matthias paper. Okay, so to summarize this one, in meta-communities with temporal variation in dispersal, or I should say meta-communities with temporal variation in dispersal have lower alpha diversity and higher beta diversity than we would predict with models based on constant dispersal. So this is an example where we took mesocosm experiments where Sarah Lee and John Bruno did manipulate temporal variability and dispersal. And we combined them with an additional statistical analysis that wasn't originally part of the paper. And we are applying this, or the idea is we could now apply this to our work in seagrass meadows in a real landscape where we have some idea of the temporal variation of dispersal potential. And I'll come back to this workflow of mesocosm to the field and this cycle later at the end of the talk. Okay, so this is what I just said. Our next steps would be to use these mesocosms with theoretical results to generate expectations for field patterns. Okay, so just to remind you where we are in the outline, we're still in this section 2, and now I have a second example of Jensen's inequality and population responses to temperature. And I know that some of you have seen this presented before, so I'll do it quickly, but I did choose to include it because it's a good illustration of a place where we don't reject the Jensen's inequality explanation. This work is from this next section is from Joey Bernhardt's research and one of her PhD chapters. Okay, so temperature is on the x-axis, and here we have a classic nonlinear relationship between temperature and performance. In this case, we'll be talking about population growth rate. And we know that in nature, most often when we're thinking about temperature, we may choose a mean temperature to plot a figure like this or to do physiological assays. But the experience of any organism or population over time is typically in variable temperatures. So there has been quite a bit, or a fair bit, a growing amount of theoretical work that has considered how climate variability could alter predictions for ecological responses to warming. And there's some really great papers on this. And I'll just reminding you that we're talking about Jensen's inequality here. So the way we can think about this for temperature is we can think about time an organism or a population would be spending in different thermal environments along its thermal performance curve. So if we think about the time spent in the thermal environment, we can think that above the inflection point of this function that describes performance over a temperature gradient, population growth decelerates with increasing temperature. So fluctuations from here and above are going to bring with it a deceleration, still positive population growth, but a weakening of that temperature effect. Whereas fluctuations that go below this inflection point, indicated here, I think nope, not by the blue line, below this inflection point, accelerate growth. So we get a different effect of dips in temperature as opposed to jumps in temperature based on where they fall on this thermal performance curve. So this has the effects of taking a thermal performance curve, as in black, that we might make in clean lab conditions and constant temperatures, as we very often do in physiological ecology assays. And it effectively shifts the performance curve with a peak performance that goes down and also moves a bit to the left along this axis. So simply experiencing varying temperatures around a mean changes the shape of the thermal performance curve. Okay, so that's our Jensen's inequality hypothesis for how population growth might differ in varying temperatures relative to constant temperatures could just be from the Jensen's inequality, which is to say the mathematics of the thermal experience. We don't have to invoke any biology at all to explain a shift in the thermal performance curve, as is shown here. In fact, we have to say that biology, whatever's going on, is not compensating for or amplifying the effect of temperature variation. The biological rate is simply tracking it in real time, and mathematically we expect a shift in this performance curve. And I hope you're already thinking, wow, we use performance curves for a lot of things, especially projecting up to biogeographic patterns and shifts with climate change. And I'll show you in a few slides that this shift can actually have measurable effects on those projections. Okay, so an alternate hypothesis to the Jensen's inequality mechanism is that time-dependent responses, so biological processes that might integrate or have time delays or compensate for temperature, would lead to different patterns. They may cause less variation or more variation. So at this moment, we just consider an alternate biological hypothesis of difference. So Joey did mesocosm experiments here, maybe microcosms. These are phytoplankton in test tubes. She grew populations at 6 different temperatures under 2 treatments. The first treatment was these 6 temperatures at constant mean temperatures of 0, 5, 10, 15, etc. And then estimated their population growth rates at these temperatures. And then she used these growth rates to use Jensen's inequality over their experience to predict what would that thermal performance curve look like if the populations had instead experienced fluctuating temperatures of the same average. So her second set of treatments was the same average temperatures, but daily fluctuations of plus or minus 5 degrees. And the dashed lines are the predictions. And in fact, the population growth rates match those predictions almost exactly. So what this means for this experiment is we don't reject the simple hypothesis that the thermal performance of population growth in this highly controlled mesocosm environment is explained by nothing more than the mathematical experience of that, the mathematically described experience of those populations. We didn't need to invoke anything else. And furthermore, we see that the important parameters or features of the thermal performance curve, Tmax, the maximum temperature of positive population growth, or Topt, the temperature at which population growth is maximum, those both shifted to the left as predicted, based only on variability. So just to sort of quickly show you the potential implications of this for the way we use thermal performance curves for studying climate change, we can start by thinking, here's a map of the standard deviation of daily ocean temperatures. We often see maps like this of the mean. Here we see a certain geography and spatial variation of variability. That makes sense given how the oceans work. And Joey used a database compiled by Mridul Thomas and colleagues of phytoplankton growth rates measured in situ in the field. And these little circles just show you where these populations were from. So she went back into this temperature data set and pulled out the thermal regimes of these different locations. And the hypothesis is then, given a performance curve of these different species under constant conditions, but then a likely field experience. So here's the average, the distribution of annual temperatures at the sites where they were collected. We can see that for most of the phenotypes, they're not actually living in temperatures that are well described by the environmental temperatures, or I should say the environmental temperatures are not directly sort of falling on the peak growth of these populations. And then we can use the Jensen's inequality that was validated by that mesocosm experiment to just fit to these data and then predict, given the variation in the thermal environment, what are the likely population growth curves over these regions. And when she did that, she found, she took into account the curve skewness of the different thermal performance curves and those field temperature regimes. And she saw that for almost every species, the static constant temperature thermal performance curve over predicted the population growth in the field. That wasn't true for all. There were a few that had right skewed thermal performance curves, and we saw the opposite. So this really matters because there's been a lot of papers that have predicted that species that have used thermal performance curves and their properties to map where species are found in nature and how those ranges might shift with climate change. And one of the sort of higher profile findings that comes out of this is this weird pattern whereby species in the ocean, species on land are overfilling their cold range limit. So they're more present there than we would expect, and they're underfilling their warm range limit. So what that looks like is that the cold range edge is not geographic range edge, is not well predicted by the temperatures. And the realized warm range edge is also not predicted by the temperatures for terrestrial species, and this was not true for marine species. So we suggest that this is actually entirely consistent with what we would predict based on thermal variation and variability. So rather than some different ecology we don't need to invoke different physiology or different ecology for terrestrial versus marine species to explain this. It could be that we simply need to just factor in variation into these projections and we would get a similar answer. So just to be clear, we haven't done this reanalysis yet. Jen Sunday is a collaborator on our variability analysis, and this is something that probably should be done, but it does provide an alternate explanation and a suggestion that considering variability for global change projections is really important. There's one more example of this that was the first one that I was part of that I just don't have time to go into, but want to point out to you. There's a sort of what's become almost a dogmatic comment with regard to biogeographic impacts of climate change, that tropical species are more vulnerable than temperate species. And that idea that tropical species are more vulnerable to warming came from analyses that didn't include variability, and they looked at thermal performance curves and global warming forecasts. And so with collaborators as part of a working group in 2014, we asked, would we get the same predictions of tropical species being more vulnerable if we considered the natural variability in temperature and the forecasts for warming? And so these three figures simply show you with latitude, we used the same data set that Tewksbury and Deutsch and Huey et al. Used to make that original conclusion. So using those same data and going back and getting the temperatures from those different sites and factoring in variability into our estimates, and then we estimated fitness, basically population growth. What you can see is there's really no clear trend with latitude in who's expected to experience the greatest fitness losses with warming once we consider variability. And in fact, the tropical species, if anything, are less vulnerable, but we wouldn't make that conclusion on so few species. And again, if we just consider different forecast response to predicted mean change or standard deviation changes in temperature, we don't recover the result that tropical species are more vulnerable. So using the same data sets, but actually factoring in variability, we get a very different conclusion. And there have been several other papers that also have suggested that the conclusion that tropical species are more vulnerable doesn't hold up when we consider variability. So variability can really change our predictions. So just to summarize this first or the second sort of the big part of the talk, I emphasized in these two examples, null hypotheses versus ecological hypotheses. And my suggestion is that the proper null hypothesis for warming for any change in variable environments might be one that takes into account the nonlinear averaging. So the Jensen's inequality, if the process of interest is actually nonlinear. The alternative being ecological hypotheses. So Jensen's inequality and other mathematical properties, they do provide simple explanations for how variation in the environment affects ecological systems. So we could characterize this as variability in the environment, either abiotic or biotic, as in the case of dispersal, simply can be explained by a mathematical accounting of ecological rate. So the rate in question, population growth rate, let's say, is more or less a passive responder to variation in the environment, and that's described by the mathematics. So sometimes these explanations are sufficient, these biology-free explanations. And so that means that's good. That's actually good news. It means we can use math and some relatively few biological measurements to generate these null expectations for biological patterns in a temporally varying world. And in those last two examples I showed you, those might already be doing a better job than the versions we were doing before without them. So we can get a long way with relatively little information. However, sometimes ecological hypotheses based on more complex dynamics, like competitive meta-communities, are more likely to explain our patterns than Jensen's inequality. So in this case, variability is affecting the relative importance of different processes. So here we really do need ecological understanding and models of it in order to understand these. So these examples, the examples of ecological hypotheses that I've given so far are relatively simple. They're meta-community dynamics or time delays in population dynamics. But of course, we can get much more complicated than that. So to sort of take a jump in complexity and sketch out how we might start to think about that, I'm going to shift our conceptual model a little bit. And now we can think of variability in the environment affects our ecological system, as we just considered. But now let's consider what happens when the ecological system feeds back on itself in response to that variability. And then furthermore, those feedbacks, that regulation in the ecological system could actually change the way variability is perceived or experienced by the ecological system. So now we're adding in not one, but two feedbacks. So it gets quite complicated, but we have some possible ways forward to guide our thoughts on this. So here's this introduction of a framework. And the timing of this is excellent. This is from a special issue that I believe was headed up by Helmut's group. I hope I have that correct. I meant to double check that. And it's coming out online November 2nd. So just in a few days, you will be able to read this paper. And this was another one led by Joey in collaboration with Jennifer Sunday and Andrew Gonzalez and myself. And it's called Life in Fluctuating Environments. And I really do love the poetry of the title, getting back to the point that life might actually exist because of variation in the environment. So I just want to sum up here in words that we're no longer thinking of ecological systems here as simply responding to the environment. We're now considering feedbacks in which the ecological systems, these could be organisms, populations, or other systems. It's more general than just the organism. These systems change the way they perceive or interact with variation. And this was our little diagram. So to start this, we just need to think of variability as a challenge. So it's a challenge because ideally, if we think of a thermal performance curve, wouldn't it be nice if organisms or populations are existing at their optimal temperature? Then they can grow fast and everything is good. But of course, in a variable environment, even if circumstances allow a phenotype to be nicely aligned with the abiotic conditions, that environment is always changing. So that's the challenge. Nobody gets to have it good all the time in nature. It's unlikely if in the case where they're always passive respondents to their environment. Okay, so if variability is a challenge, predictability is a solution. So you can imagine the benefits. So there's costs perhaps to not managing one's experience in a variable environment, which could mean that there are often benefits to prediction. Prediction meaning, so how, there's so much to say here and not enough time. So I will simply point out that from a time series like this one, there are many different ways that an organism or a system could predict the pattern in this time series. Spectral analyses can allow us to look at the possible opportunities for prediction or sort of highest predictive parts of a time series. And different organisms with different perception abilities or different sizes or different experiences of the environment may actually pick up on different parts of a frequency, of a pattern of variability. So there's lots to explore here, and this is one area where mesocosm experiments could probably keep us busy for a very long time. But there's also lots of, here's sort of the scaffolding of our framework, and I hope this is something we can come back to in our discussions. So I've mentioned, if we consider a cartoon version of variability here, fluctuating environment up and down of, let's say, temperature or something, we've already considered something like a passive response, which might be the biological system mirroring exactly the environment, or a filtering or sort of a slightly dampened experience where the biological system is always responding to the environment. A second, though, category of ways to think about variability and responses is when we allow biological systems to sense themselves and have homeostatic feedback, such as thermal regulation. So now the organism in this case might be sensing the change in temperature and then reacts when it senses its own body temperature change, and then initiating physiological mechanisms that allow for regulation of body temperature. So in this case, the system's not necessarily responding to the environment, it's responding to a change in itself. A second class now gets into what we call, or what are called, anticipatory feedback. So now a biological system can not only sense itself, but it can sense the environment, and it has learned through some way, evolution or other processes, to link environmental conditions with a likely future state of itself. So in this case, the organism might sense the change in temperature. That might serve as a cue indicated by this asterisk, so that the organism already starts its response to what it knows is going to be a likely future condition. So this is initial, if you haven't thought about it, it can be an uncomfortable idea because we're very well trained that organisms can't predict the future. But there are many, many examples throughout biology actually of anticipatory responses, feedbacks, and the phenological cues are just one of them. And then a third class would be general adaptive systems that includes both of these classes. So I don't have time to go into that more in this talk. I encourage you to check out the paper, and I hope we can discuss this more. And Joey is one of our discussion leaders for the discussions after the talk. And so if you're she's definitely someone to talk to more about this. So the last few words I would like to say is just back to our topic of mesocosms as tools for generating expectations for changing ecosystems. So I often use this figure to sort of capture practicing science in a changing world. And on this figure is scientific understanding and expectations. So this is where mesocosms come in. They play a role in helping us figure out how we think nature works. And then in our science or in our collaboration with others, these things inform how we go out into nature and measure the environment, measure biodiversity, measure growth rates, whatever it is. And then hopefully they also are they should be influencing the way we analyze our data, what we infer, all the way to detection and attribution of change on the ground. And it's detection and attribution that really make it into these assessments. And it's the assessments like the IPCC, IPBES, or regional or local assessments. It's these that influence science-based policy much more than our mesocosms do. So in my experience, our mesocosms are part of this bigger process that, if well connected and done right, our mesocosms are then indirectly affecting science and policy and understanding through this filter. So this is important because it really focuses where we have the most power with mesocosms. We could also, you could draw other arrows here to think about the relationships on this landscape for sure. There's two ways I want to mention, and I see this a lot, and I've done this in my own research. We can think of, the first one is mesocosms could be considered simulations of the future. So we see this a lot in how warming or acidification experiments are presented. We have an ambient or a present day treatment, and then we have a +3 degrees or a plus a certain level of pH increase, with the idea that that is in effect a simulation of a 100-year future scenario. So these, I call them simulations because often we don't have great theory or predictions for what we're going to observe, and we're using the experiment to actually maybe help guide our understanding. So it's almost like, it's a simulation of what we think will happen that then we're going to try and understand. And then we're going to take this module of scientific understanding, put it back in that bigger process, hopefully. So the advantages of mesocosms as simulations are they can help us explore consequences of experimental treatments like pH or temperature or plastics, where we don't actually have theory. So we can use them when we lack a well-developed theory to test. And I have an asterisk here because this isn't always a great idea, but it is how we use them, and sometimes it is a good idea. The disadvantages of this approach, however, are important, and that's without a theoretical framework to inform how we can apply the findings across scales, the inferences that we make from simulation type mesocosm experiments cannot be scaled up. Or the inverse of that is to say, if we extend beyond the scale, the treatment levels, and the time span of these experiments to broader scales, we're doing so very much at our peril because we're lacking the very theoretical frameworks we need to tell us if that's okay. So there are some real dangers with mesocosms as simulations, even though sometimes they're all we can do. The alternative is mesocosms as tests of theories, and this is where their real power is, in my opinion. The advantage is that we can cleanly test critical hypotheses that come from larger, more complicated theories. And this is possible because in mesocosms, of course, we can control many, many factors that we just can't control in nature, like temperature. The other advantage is that we can use empirically, they can be used to empirically estimate parameters. So again, if we have a model, a simulation model or a theoretical model that we want to use to forecast change, we can use mesocosm experiments to get some insight onto how we should parameterize those models. That's another great use of mesocosms, and one that perhaps is sometimes overlooked or undervalued, but I think it's really important. And in fact, this one is often how mesocosm experiments enter into something like the IPCC reports, specifically as parameter validation. So another advantage is that the theoretical framework that we use allows us to scale up from the mesocosm, because, as we know, most mesocosms don't include dispersal. That's a major limitation of nearly all mesocosms. However, our models can include dispersal based on other experiments, and then together those things could allow us to make projections for a larger scale where dispersal is definitely happening. The disadvantages of mesocosms as tests of theories is the same one I've mentioned. Theory is sometimes lacking, or sometimes it's so complicated that we can't, it's too hard to figure out on our own, or we lack the collaborators to actually figure out how to test the theory, or the theory is done in a way that simply is not testable. These are all problems we have in ecology right now. The disadvantages also of mesocosms as tests of theories is they are necessarily simple. To do this work, to get the advantages of mesocosms, we have to keep them simple, because once we get more complicated, and I've done it, I think everyone who's done mesocosms has done it, you add in one more thing and then suddenly you get results that you don't expect, and it's very difficult to interpret, and now it's harder to achieve our goals. So one of the resolutions here, I'm a huge believer in mesocosms. We definitely need to keep doing them. And I really love collaborating with theoreticians and modelers on mesocosms, because often they help me understand what I'm seeing. So how can we use mesocosms to make better projections about change in a variable world? Well, this leads us into our discussions, and the discussion leaders did give me some ideas of what they're talking about. And I just picked representative questions. Apostolos will be talking about how we, among other things, suggest we should be considering how can we expand our variance explicit approaches to multifactorial and multi-stressor contexts. This is a very important question, challenging the limits of our mesocosm experiments. Sam Fay is going to talk about what are the consequences of organisms experiencing different previous environments, so integrating that history in a varying environment to understand the present experience. And then Joey suggested another important question is really getting an understanding of what mechanisms do organisms in your system use to deal with variation in their environment? And are they passive responders, or do they have a way of predicting the change? Okay, so really I just wanted to close with this idea, the challenge of integrating variability more into our thinking, and doing so and taking the most advantage of mesocosms, which are, I think, essential tools to this process. And often mesocosms aren't always at the forefront out here, but they're necessary here, and we can make sure that they're better integrated into this process. Okay, so thank you to the people whose research I shared in this talk, John's and Joey's, Jennifer Sunday's, Sarah Lee and Adrian Steyer, as well as our whole group of collaborators on those projects. And thank you for your attention. I look forward to questions and discussion.
Thanks a lot, Mary. Yeah, sorry, Helmut.
Sorry, Marie, go ahead.
Thanks a lot, Mary, for this wonderful talk. And we have some questions in our question and answer. I don't know who started first, but I will start with one question. So why opposing math in biology? Jensen's inequality assumes a type of biology, for example, in organisms, the absence of acclimation and stress responses.
Yes, that's an excellent question, and I completely agree. I think what I mean by that is when we make our hypotheses, until a few years ago, we weren't thinking about Jensen's inequality, and we were all reaching for biological hypotheses in the sense of sort of more complex mechanisms, like you say, acclimation, adaptation, plasticity. And I think we don't need any of those to explain these patterns. But you're right, that still is biology without invoking those mechanisms as the driving explanations. Does that make sense?
Yes, thank you. Thank you. And I will just continue with the next one. So it says, many thanks for a great and informative talk. I want to ask you about genotypic adaptation as a source of variability. Mesocosms are often assumed that species are static in relation to abiotic stressors like the thermal response you showed. Yet we also know that local adaptation is common in species, but it's also a source of variability. Do you think there are ways to account for this adaptive variation in mesocosm experiments?
Yeah, that's a great question. And there's Matt Siegel did research in our lab that I didn't have time to present that looks at that. And I can just, there's many ways to do it and many people are doing it. A couple of ways are to include different clones. So you could explicitly include the potential for different phenotypes in your systems. And there are experiments who have crossed clonal diversity with different treatments. So we could do that with variability. I know I've seen people suggest this experiment. I don't know if it's done yet. Another possibility, one thing that Matt did is he used the tide pool copepod Tigriopus californicus. And these are they're copepods that are found in the ocean, but they do thrive in high intertidal tide pools and they can get isolated from larger populations. So other people have shown a really high potential for local drift anyway, maybe adaptation, maybe drift to in these different tide pool populations. And Matt collected a bunch of those and exposed them to heat waves with the hypothesis that different, so this is blending adaptation and possibly the possibility of different thermal backgrounds from those tide pools to see if they responded to heat waves differently. And in that case, we were surprised we didn't actually see differences in response to heat waves, but I think there's no reason that has to be the general result. It could be different depending on what's driving adaptation. So I think that's a great question, and there's many ways that people are exploring it and that can be explored further.
Okay, there are many more questions. Miriam, do you want to continue?
Yeah, I can maybe do the next one then. Yeah, Jens says, when you concluded that the small mesocosms experiment in low, medium, and high supply rates did not respond in a way that supports Jensen inequality, is it possible that all the treatments had a relatively high supply rates and thus was all out on the right side of the Jensen's curve? Some things in nature may work over much longer time and at lower than many of experiments covers, right?
Yeah, absolutely. In that experiment, and I don't remember off the top of my head the exact dispersal rates that Sarah used, but the low was quite low. So in that case, over that time period, the experiment went for more than a month. I think it's unlikely we're missing the low end of dispersal and our response, of course, is those relatively short-term population responses. So maybe we got a one generation time of some of those amphipods, maybe two, but certainly not more than that. So obviously there's no effect we couldn't talk about an effective dispersal on any sort of genetic changes. And that's a very important role that dispersal plays in nature and variability in dispersal. And we couldn't even really talk about the potential for longer term extinctions and population dynamics. So you're right. I certainly wouldn't use those results to reject Jensen's inequality as an explanation for dispersal variation in the field or at broader scales. Nonetheless, we were actually a little bit surprised we didn't see it even in that experiment.
Yeah, the next question is about should we think about temporal autocorrelation within the framework you've presented? So will it change the shape of the thermal performance curve? Likewise, he mentions that changes in the color of noise could undermine the predictive mechanisms that organisms may have.
Yeah, excellent. So those are two things that come up that we wrestled with a lot and offered a few thoughts on in that philosophical transactions paper on the life in fluctuating environments. And I just didn't have time to do that here. But you're absolutely right. So autocorrelation comes in one way in which it comes in is it lends predictability to variation in the environment. So if organisms can detect, or if there's selection for performances that reflect that autocorrelation, and that's probably something that's going on in seasonality. We probably see those effects of autocorrelation in nature much more often than we realize, only because many of us haven't thought about it much till recently. How it changes the thermal performance curve, actually, that's a great question. And I can't answer that off the top of my head, but I suspect people here collectively could. Yeah, okay, I think that's all I have to say. But those, again, great points. And I guess in moving forward with variability in mesocosms, some of those things like spectral analyses have been done on field data with then hypotheses for how different variation time series affect biology. But what I haven't seen much of, if any, is mesocosm experiment. Well, that's not true. There are a few mesocosm experiments that really test those hypotheses about color of noise, but that's an area where it's a handful. So there's a huge potential now with our with all the technology we have in our labs now to detect, to manipulate variation and to measure high temporal resolution. I think that's a really wide open area of research that we'll see a lot more of in the next couple of years.
Okay, thank you. The next question is about what could cause species to evolve left or right skewed thermal performance curves, and how does this difference in skewness impact responses to warming overall, especially in the tropics?
Ooh, good question. I cannot explain why we see those different skewness. This would be a question for Joey, if you're able to follow up with her. I don't know if she's part of the panel now. Can she answer that? Miriam, is that possible?
She's not. I don't know, Katharina,
okay.
If you can add someone from the audience.
Yeah, wait a second.
And you might need to repeat the question since maybe she didn't realize I was going to punt it to her, but I feel like she will have a good answer to this.
I'm here. Can you hear me?
Yeah.
Okay. Hi, everybody. Yeah, great question. The short answer is we don't know. Lots of people are wondering it. All I can say is that there are some examples of right-skewed performance curves in parasites, but I don't know more than that. We don't actually have enough examples that at least we had in this data set where we could derive any sort of generality to what factors are associated with those right-skewed curves.
Thanks, Joey. And I would just like to say, Joey saying she doesn't know is different than me saying I don't know. That's not a question I'd asked myself before, but I know it's something she's asked herself. So that I don't know, I think means that it's something that people are working on and should be working on.
Thanks to Joey and Mary for this question. And we have more questions. So Daniel Petfield says, great talk, Mary. Just wondering how your life framework fits with types of variation. For example, what responses would you expect with random variability compared to expected variability? And he also adds the question, also over what timeframes do you think variation is important within an organism's lifetime or across generations?
Yeah, great question. So one of the things that I'm excited about with this framework is it allows us to start thinking about organisms or communities or populations or any level of organization you want. It allows us to ask, okay, in the immediate sort of temporal vicinity, an organism is responding to its environment. But over longer periods of time, these systems may have either evolved or self-organized in ways that actually reflect environmental variation regimes. So their size and their network various networks for resources and communication, and their phenology also reflects that environment in such a way that might be a solution to essentially a way to minimize the experience of variation. So to me, a question like yours, I think about it now a little bit different. I mean, there's a practical answer like, okay, I want to know about this population now, then yes, I need to think about the last 10 years, 100 years, and the different phenotypes. But about ecology in general and global change in general, if, and I'm not sure we know this yet, but if we really are changing variability regimes, we might be changing the size and the organization of living systems, because if those things actually reflect variability, they may be able to adapt. They may adapt by breaking into smaller parts, by joining into larger parts. They may adapt by changing in ways that we perceive as change, but could in fact be system level responses to a shift in a variability regime. So there was one other thing to your question, but anyway, I guess I would approach a question like, what's the right scale? If you have the luxury to think bigger about how the biosphere and nature is working, then I would take it from that perspective. And we tried to consider this in the LIFE paper, as well as different random versus regular variability regimes. And if you need to solve a practical problem right now, then I guess you kind of have to leave that larger organizational question aside, but still be open to the fact that reorganization could be the solution to changing variability. It might be a type of change we need to accept and perhaps even foster and encourage, because that's where the resilience in the future may come from. And the last point, right, you asked about regular versus random variability. Well, in consistent with my answer there, random may be random at one resolution, but not at another. So an easy fix is for an organism to you know, I don't want to use the word evolve. It doesn't always have to be evolution, but to shift in some way to experience the environment at a different resolution, maybe by hibernating, maybe by only being active at certain times, maybe by changing body size, bigger or smaller, maybe by becoming a symbiont, becoming a parasite. You know, there's all kinds of ways to change the way you experience the environment, and that may move you out of random noise into predictable variation.
Okay, thank you. The last question is more maybe a comment. It's about that it's true that most mesocosmos may not lend themselves to dispersal studies, but there are few being built. Perhaps we need more of this?
Yes, I really think so. And especially, you know, one thing that we're working on and have, an area of our lab's work right now is this idea that almost every, not every, but almost every mesocosm experiment on temperature has been in a closed system. So from this idea that, oh, maybe adaptation, you know, either through actual evolution or through replacement of phenotypes and genome sorting and things like that, we haven't allowed those possibilities over thermal gradients in our experiments because we've kept them closed, because that's really how you have to do mesocosm experiments. But I'm really happy to hear that, and I know that there are many groups that are starting to make dispersal implicit in those designs, and I think that's definitely got to be the next step.
Thank you, Mary, for a really inspiring presentation. And you are right, the special issue in Philosophical Transactions, where your live paper with Joey and Jen and Andy is published, is a outcome of a HIFMB symposium 1.5 years ago, where we were still able to meet in person. And with that, I would like to close this session, say thanks again for your great presentation. And I would like to pinpoint that the webinars of AQUACOSM are recorded and available on the AQUACOSM webpage. And also the World Cafes, which are happening afterwards, they are summarized, and also these summaries will be there available soon so that you can see what type of discussion about the future of mesocosm research has been discussed in this consortium. So with that, I think we are moving towards the World Cafe phase. Is there any announcement to that from Katharina or someone else?
Thanks, Helmut. No specific announcement. The people who are joining the World Cafe have received the links. And yeah, from our side, from the organizers, thanks again to Mary O'Connor and to the group in Oldenburg for organizing this wonderful webinar and for this inspiration. Thank you.
Thank you.
Bye.